video we are going to talk about translation. We've already done the preceding videos, and now we're going to talk about translation. So a quick recap of the central dogma. We had DNA forming mRNA forming protein. This was transcription, and now we do translation. Okay, so before starting translation, we need to prepare our stuff. So it is just like cooking. We need to prepare all our ingredients before we start cooking. Just like when you make fried rice, you have everything ready on the table, your shallots cut and chopped, you have your sesame oil or your any kind of oil, and your 1-day-old rice. Like that, you have your counter set. So we need to set our counters as well. So the things we need for translation to run smoothly, we need good transfer RNA. We need ribosomes. Ribosomes can be free. It can be freely floating in the cytosol or attached to the rough endoplasmic reticulum. And we also need messenger RNA. We need a lot of protein factors. And we need energy. We need GTP and we need ATP. Right? So all of these stuff we need for translation to occur smoothly. Now I'm going to explain what is charging. So how do you charge your transfer RNA? So now we are gonna talk about charging. So this right here is charging. This whole stuff from here. So we have an amino acid and then an enzyme denoted as E, amino acyl tRNA synthetase. It is going to have require ATP to form EAMP amino acid. And then this EAMP amino acid, our tRNA is going to enter, and AMP is going to leave, and E is going to leave. And on this area, on CCA, our amino acid is going to get attached. And at here, at this point, our amino acid is going to get attached here. And at this point, at this here, this is the anticodon loop. Here we are going to have our anticodon complementary to our codon. See? So like for example, we have AUG, that is our codon. What is going to be its counteractive, is complementary. It's going to be UGA. Sorry, UG CAU. UAC is going to be our complementary anticodon. So as you know, this was 5' to 3'. This is going to be anti-parallel. This is going to be 3' to 5'. Okay, that's that. And this is our charged methionine. This is our charge. You see, this was AUG, right? It was AUG. UAC, it was, complementary, and then there was methionine. How does this happen? Now, I'll tell you one thing. The transfer RNA is going to get charged with amino acid first, and then it is going to find its code on the mRNA. It's like the same example. Now I'll give you an example. For example, I'll just draw an example here because that's more interactive. As you cannot see me, you can just see my screen. You see, if there is one man, this is man, let's see, this man is transfer RNA. And this man has mangoes. So these are mangoes. And another man has peaches, and it's another tRNA. Now these, they have mangoes and these have peaches, and now they're gonna look for people. I'll make people with square heads now. They're gonna look for people who need mangoes, who need peaches. For example, person A, he needs peaches. They're gonna bond with that. Person B needs mangoes. They're gonna bond with that. So the transfer RNA is first going to have the mangoes or the methionine, and then it is going to look for the start codon. Okay, that is the AUG, and then it is going to get attached to that mRNA. Okay, now there is another interesting hypothesis called as the wobble hypothesis. Now what does wobble hypothesis mean? Is that I told you about the example of mangoes and peaches. Now what actually happens is that first I'll tell you the case, and then I'll give you the example. What happens is in wobble hypothesis, and then this is your mRNA, and this is UAC. Okay, this is UAC. Now we know that, for example, this is just an example. If you have UAG, this is a stop codon. We're not going to talk about this. Let's change this. Let's do this GAC, and then we have the transfer RNA that is complementary, like G, U, and C. So let's see if it does not have G, it has A, right? So this is located within our transfer RNA. Okay, this is located with our transfer RNA, right? Now you see this is complementary to this is complementary to this, but this is not complementary. But according to wobble hypothesis, the transfer RNA is not going to be concerned about the last base and still gonna get bound to this mRNA and get that attach that little molecule, the amino acid here. This is called a wobble hypothesis. It is going to very much careful about the first base, about the second base, but not so much about the last base, right? So like an example right here, if this man wants mangoes, like this A man wanted peaches, but this man comes here and say like you kind of like mangoes too, so I'm gonna give you a mango too. So they will give mangoes to A, right? So that's like wobble hypothesis. The importance of wobble hypothesis is that in previous video I told you about silent mutations. These silent mutations are gonna get corrected, even though if there is another kind of point mutation as well. That can be avoided if transfer RNA does wobble hypothesis. Okay, let's move on towards initiation. Now for initiation, there are two more things that we need to know. There are slight differences between the prokaryotes and the eukaryotes that we need to talk about. First is the Shine-Dalgarno sequences. Now the Shine-Dalgarno sequences, they are only in the prokaryotes, and they are basically like, why are we doing this? We are doing this because we need to start somewhere on the tRNA has to look for the start codon, or the ribosome has to look for the start codon. How do they reach the start codon? How do they recognize the start codon? That in prokaryotes is Shine-Dalgarno sequences. This means that the 30S subunit of the ribosome has some 16S RNA, which has some upstream to it some sequences. Like for example, this is the 16S ribosomal RNA, and upstream to it, like at the 3' end, upstream Shine-Dalgarno sequences are present. They are called Shine-Dalgarno. They're just like random AU stuff, or GAU, GC stuff, and that is basically complementary. complementary to the mRNA. It is complementary to the mRNA, and they bind, right? And they bind, and then they recognize AUG. So they're like 20 to 30 bases up the mRNA, the Shine-Dalgarno sequences, and they are complementary. The Shine-Dalgarno sequences are on the mRNA, I'm sorry, not on the ribosomal RNA. They are present on the mRNA. SD are present on mRNA, and then the 16S are complementary to that. That's Shine-Dalgarno. And then we have in eukaryotes, we have the beautiful 5' cap, and the 5' cap, they recognize the 5' cap by the 4'A subunit of the eukaryotes, and with the help of IF4 proteins, they're gonna then they're gonna scan, basically the happening of scanning, and they're gonna reach to the start codon. Okay. Now, recognition of the AUG, one after that, after the above information, is the initiator transfer RNA. How does the initiator transfer? This was how the ribosome is going to recognize, and this is how the initiator transfer RNA is going to recognize AUG. It's in prokaryotes by IF2 GTP, and in eukaryotes by EIF2 GTP, right? So you need to remember these all. Now, only in prokaryotes, the initiator transfer RNA actually had to undergo transformylation. What does that mean? Is that we have this methionine initiator transfer, this is charged transfer RNA, and we have the N10 formyl THF tetrahydrofolate. It gives its N-formyl group, and then the N10 formyl group attaches with the N-formyl methionine, and it forms F-met tRNA. Only in prokaryotes, it does not occur in eukaryotes. Now we start our translation process. Now what happens, I'm going to zoom in this diagram. So the first is our charge transfer RNA is going to sit in the P site on the small subunit, and it is going to look for AUG. So initiator factors, they form the initiation complex. So this right here, this whole is the initiation complex, and the charge RNA is going to sit on the P site. So here is the P site, this is the A site, and this is the E site. Now let me show you in the large subunit, that's better. This is the A site. The A site is mostly where a new charged tRNA is gonna enter. The P site is where the magic happens. It is the prime minister seat, and the ME site is basically the empty site. Everything is gonna leave here. Okay, now this is the large subunit. Now what happens in the step 2 is that this large subunit is going to sit on top of the tRNA on the small subunit. Now this whole is an active ribosome. Now initiation factor 1, 3 and IF2 GTP is the GTP is going to get hydrolyzed. So we use the GTP here. The first GTP is used here. Yes, the first GTP is used here, and then our active ribosome is now set. Now in the third step, a new charged tRNA with phenylalanine is going to enter, and when it enters, elongation factor EF2 GTP, it will bring the charged tRNA, and when the GTP is hydrolyzed, it is going to sit on the A site. You see, it is sitting on the purple color, so it will sit on the A site with the help of EF2 GTP when it hydrolyzes. Okay, now the next, because it is also corresponding to the mRNA, bear that in mind. Now our third step is our big step, that is transpeptidation. What will happen is that the peptidyl transferase, it is present in the large subunit, it will form a bond between F-met and phenylalanine. And when this bond occurs, this peptide bond will form, Also what happens that this phenylalanine, it will shift from A site to P site. You see here it has been shifted from purple to blue, from A site to P site. This is called, remember, the transfer RNA is not shifting, only the amino acid is shifting. Okay, only the amino acid is shifting. This is called as transpeptidation. Okay, cool that. Now what will happen, the next step is translocation. Our fifth step is translocation. In translocation, elongation factor GGTP will be hydrolyzed, and it will cause the ribosome to move. Now the ribosome will move along the RNA, it will move along the mRNA, and it will move everything, the tRNA. Now everything that was in A site will be moved to P site, and everything that was in P site will be moved to E site. This is translocation. So you see here the transfer RNA, which had phenylalanine, is now from purple to blue, and the F-met RNA is now from blue to green on the empty side. The next step will be that this F-met tRNA, it is going to leave the ribosome via the empty site. Now sixth step is we will repeat these steps, this transpeptidation and translocation over and over again, and we will have a polypeptide. And how does termination occurs? In termination, the termination codon is recognized. So RF2 and RF1 will recognize UAG, UAA, and UGA. Right, it will recognize these, and RF3, it is going to hydrolyze GTP, and the RF2 and RF1 is going to release the transfer RNA from the ribosome, and when that happens, the large and small subunits are going to dissociate, and the polypeptide is going to released, and that is how translation occurs. Now one last thing before we wind up, there are certain covalent modifications for the polypeptides. We can have phosphorylation, we can add phosphate group, there could be glycosylation, we could add glucosamine or glycosamine, we can hydroxylate, we can add hydroxyl group, or biotinylate enzyme we can add, this is the group. We can carboxylate the polypeptide, and we can add the farnesyl group as well. So different covalent modifications according to different, like for example, I can give you an example of hydroxylation. The collagen protein is hydroxylated at the sites of proline after when the polypeptide is formed. Just an example. And one more thing before we end is that most of the regulation occurs at the level of transcription, but regulation can also occur at the level of translation if we modify elongation factors. Also, one more thing you can do in modifications is you can trim the protein. So protein trimming, you can cut the protein as well. So these were some things that were important, and I will see you in the next video. Thank you so much.